Okay, hello everyone. Welcome to the webinar today. I'm going to go ahead and get started. I think probably will be a few people still trickling in. I'm Stephanie Shadbolt, the Outreach Coordinator for MIDAS, and I'm hosting the webinar today. This is the third webinar in the MIDAS webinar series, where we feature current research by MIDAS members. Just so that you know, the webinar is being recorded and the video will be available later on in the MIDAS website on our events page. So for those of you who may be new to MIDAS, MIDAS is a global network of researchers, students, and others who are dedicated to the modeling of infectious disease dynamics. MIDAS membership is open to anyone who is working in or has an interest in this area. And you can find the membership application form on our MIDAS, our website at midasnetwork.us. And of course, if you're looking for general information about MIDAS, you can also visit the website to learn about the mission of MIDAS, who the members are, their research projects, the COVID-19 portal, as well as news and events. So now I'm very honored to introduce today's speaker, Dr. Micaela Martinez. Micaela is an assistant professor of environmental health sciences at Columbia University. She is an infectious disease ecologist who specializes in the transmission of acute infectious diseases. Micaela completed her undergraduate training in mathematics and biology at the University of Alaska Southeast and a PhD in ecology and evolutionary biology at the University of Michigan. Her ongoing research falls into four themes: seasonal disease transmission, maternal immunity, the realized effects of vaccines, and interactions between biological rhythms and disease. Micaela is the recipient of an NIH Early Independence Award, where one of the goals is to characterize functional seasonal rhythms in the human immune system. Her lab is also researching the immunity, transmission, and racial inequalities in COVID-19. Most recently, Micaela and her collaborator received an award from NSF under the Rapid COVID-19 Research Program, looking at air pollution and Google traffic data to assess the effects of social distancing. And in today's webinar, Micaela will be discussing other aspects of her ongoing COVID-19 research. So before I hand it over to Micaela, I'll just mention that we'll have time at the end of the presentation for questions. You can use the raise hand function in the participants panel, and we'll unmute you so that you can ask questions directly, and that'll make it a bit more interactive. Or you can also use the chat window to ask questions. Okay, so I'll stop sharing this screen and hand it over to Micaela. So please go ahead, Micaela.
Great, thank you for that, Stephanie, and good morning or good afternoon, depending on what time zone you're in, to all of my colleagues that have joined. So let's see if I can get my screen share started. Here we go. Okay, great. Can everybody see that? Stephanie, can you confirm?
Yes.
Okay, great. So today I wanted to talk about work that myself and colleagues, people that many of you may know. This is a collaboration between my lab, Andrea Graham's lab at Princeton University, and Andrew Yates' lab, who was previously at Glasgow, now he's at Columbia, and then also the lab of Natalie Riddle, who's at the University of Surrey in the UK. And among the four of us PIs and two wonderful postdocs, Jackie Lang and Sinead Morris, we have spent the last few months really trying to wrangle a lot of the data and information that's been coming out from the immunology literature on SARS-CoV-2. And so this talk today isn't going to be a traditional research talk, rather it's going to be a review of the state of the knowledge coming out of the immunology literature on SARS-CoV-2 immunopathology, and really trying to pose some questions for the disease ecology and modeling community in terms of how we can start thinking about immunopathology and the good and the bad, essentially, of the immune system and within host dynamics for SARS-CoV-2. And so, as many of you know, we have now over 9 million confirmed cases worldwide. Just within the last 24 hours, the CDC has released estimates that over 20 million Americans have been infected with SARS-CoV-2 to date. So the confirmed cases that we have are just the tip of the iceberg. And with the SARS-CoV-2 and also infectious diseases in general, there's this spectrum, and let's see if, hopefully you can see my pointer, but there's this spectrum in terms of infection where some individuals are completely asymptomatic, and then you can have mild cases or mildly symptomatic cases, and then symptomatic cases that may follow various trajectories in terms of outcomes. And so when we think about this in terms of SARS-CoV-2, we have some individuals that are presenting no symptoms whatsoever, others who may have fever, cough, and then we have individuals that end up hospitalized and on ventilators, and we'll talk about this a little bit more, and succumb to disease-induced mortality. So what I want you to keep in your mind throughout this presentation is that the path that an individual follows in terms of the symptoms and their disease progression, one of the main questions of interest is what is happening within the body that dictates what path you're going to follow. And as I said, these trajectories can range from being mild and staying at home or being hospitalized. But this really is an underlying biological process that's happening within the body and under the hood that's dictating these outcomes. And just to kind of frame this in terms of infectious diseases more generally, thinking about this spectrum and disease outcomes, I wanted to just share kind of some numbers from diseases that are very well studied. So for instance, for poliovirus, which is an infection that I did my PhD on, 99% of infected individuals are not showing any symptoms. It's only 1% of individuals that are on the symptomatic end of the spectrum. And then among those symptoms, you can have transient paralysis. I'm sorry, I live in New York City, and so you will hear ambulances come by on occasion, and I'll pause for a second. So for polio, in the 1% of individuals who show symptoms, some of them have transient paralysis, and they'll have recovery of motor neurons that were lost during the infection. But you'll have some individuals that experience permanent limb paralysis or even lung paralysis, which is what would end children up in iron lungs that you would see photos of from like the 1950s before we had eliminated polio. And through much of the world. And then in terms of the cases, 2 to 5% of the cases, which were just asymptomatic cases, would result in death. Now for measles is on the other end of the spectrum in terms of asymptomatic to symptomatic ratio. The majority of infections are symptomatic, and then those symptoms range from rash, pneumonia, some individuals will have encephalitis, and then deaths are 2 in 1,000 cases. And then if we look at varicella, which is a virus that causes chickenpox, similar to measles, majority symptomatic, and the symptoms can range anywhere from rash to encephalitis and meningitis, and a low death rate of 1 in 60,000. And then for SARS-CoV-2, here is where we're still trying to get this picture. The picture of what percentage of individuals are asymptomatic versus symptomatic, and then the plethora of outcomes on the symptomatic edge of the spectrum. But I wanted to highlight the importance of understanding not only the asymptomatic to symptomatic ratio, but then the outcomes using some of the numbers from polio, and why there's been so much confusion among the general public and in the press in terms of how deadly SARS-CoV-2 has been. So for instance, if we use the numbers from polio, if you look at the case fatality rate for polio, 2 to 5 deaths in 100 cases. That seems like a very extreme mortality rate. But then when you take into account the fact that only 1 in 100 infections are symptomatic and reported as a case, then what you actually come to find is that the infection fatality is 2 deaths in 10,000 infections. So this highlights that we really need to pay close attention to the asymptomatics when considering how deadly an emerging infectious disease is. And now, if we do some back of the envelope calculations, I did this a couple days ago when I was putting together this presentation. Now CDC has done, just released a more formal version of this, but as of May 20th here in New York City, or I'm sorry, May 2nd, 2020, here in New York City, the governor's office had run a serology survey, and they collected blood samples from individuals that they considered representative of the population. And what they found from this was that 19.9%, so almost 20% of individuals had been infected by that time. And so this means that it takes 2 weeks for our bodies to start producing antibodies. So if we count back a couple weeks, then we can assume about 19.9% of the population was infected by April 18th. And we can simply go through and multiply that out by how many people live in the city to estimate how many individuals will have been infected by that time. And by that week of April 18th, we had 146,000 confirmed cases and 15,000 deaths here in the city. And so what this tells us is that if these serosurveys are representative, then only about 8.7% of the actual infections in the city were identified and reported as cases. So if we use these very rough numbers to fill in this spectrum here, then that would suggest that 91% of infections are undocumented and 9% are reported as cases. And this is very similar to what the CDC has reported in the last 24 hours nationwide. Now, the percent of cases that are, the percent of asymptomatic cases that are reported is solely a function of how we do testing. If we randomly tested every individual in the population, or if we just tested every individual in the population on a regular basis for the infection, then we would pick up a lot of those asymptomatics. And so here I just wanted to show you how testing effort has changed through time in New York City. So in blue, these are the daily tests. In yellow are the confirmed cases, or the test positive. And as you can see that in recent weeks, we've had a very, very large increase in the number of tests. And so as testing occurs as a result of contact tracing, and now we have free testing for everybody, even potentially asymptomatic people, we will potentially start shifting these numbers in terms of what percent of the asymptomatics are actually captured in reported cases. Now, there was a really great report that had been put out by CDC that looked at 1.4 million cases that had come into CDC by May 30th. And so what they did was they went and reviewed all of these cases and reported a bunch of information in terms of symptoms, age, race, various demographic parameters. And one of the things that came out from this report is that only 4% of all reported cases had been asymptomatic individuals. And so that's quite low. So what that's telling us is, at least through the end of May, in the United States, what was happening, the process that was happening for observation of SARS-CoV-2 is that you have a large pool of infected individuals in the population. Only a subset of them are going to become symptomatic based on how the infection runs its course in that particular individual's body. And then a subset of those symptomatic individuals will be reported as cases. So for the most part, we have very little information in terms of the asymptomatics. But now just thinking about the symptomatic individuals, so some of the other numbers that CDC has reported is that 14% have been hospitalized, 2% have been admitted to ICU, and 5% have been reported as deaths. And there was also a report in JAMA that had reviewed 73,000 cases in China, and they found similar numbers. In 14% of cases developed severe pneumonia, 5% developed respiratory failure, septic shock, and multi-organ failure. So that 5% with respiratory failure, septic shock, and multi-organ failure is similar to the reported percent to have died in the United States. Now, the thing that our group has been thinking quite a lot about is what triggers respiratory failure? What triggers the most severe cases? And so for COVID-19, the primary cause of death is ARDS. And ARDS is acute respiratory distress syndrome. And hyperinflammation and cytokine storms are likely to contribute to this. And so this is damage done to the lung by the immune system. And we'll get a little bit more into that in the coming slides. So what is ARDS? So ARDS is results of lung injury. So ARDS is something that not only can happen in infections, but also when people experience trauma, like car accidents. So ARDS occurs when the lung has been severely injured. And what happens is that fluids start to leak into the alveoli of the lung, where gas exchange typically happens. And when there's fluid in these little air sacs, then gas exchange cannot happen efficiently. And because of this, the body becomes starved for oxygen. And this is why individuals who are experiencing ARDS in COVID-19 have very low blood oxygen levels. And in general, not just with COVID-19, but overall, ARDS can happen when an individual has sepsis, so their immune system is attacking their tissues. It can happen when they have pneumonia, and like I said, it can happen from other acute traumas. Now, the standard of care in the United States for ARDS is mechanical ventilation. And so with mechanical ventilation, what happens is an individual will have a tube that's inserted into their airway, and the ventilator will blow air or air that's enriched in oxygen into their lung. And so this is a supportive care. It's not therapeutic. It's not healing their lung in any way. It is simply trying to increase oxygen delivery to their body to mitigate the amount of tissue damage that is done from oxygen deprivation. And so because mechanical ventilation is not therapeutic, there are actually a very high percent of individuals that are put on mechanical ventilators die from COVID-19. So according to the Associated Press, as of April, 80% or more of coronavirus patients in New York City that were on ventilators died. And so there's a very high mortality rate, and this has led some doctors to start to evaluate or question whether or not this is the best form of treatment. And so just to be clear, when we think about this mechanical ventilation as being supportive care, it really isn't all that different from, let's say, individuals who were put on iron lungs for polio. It is a way to maintain life, but because it is not therapeutic, it's not necessarily assisting in the healing process. And so data that have come out over the last several months have been used to draw several timelines in terms of when individuals end up in hospitals, when ARDS typically happens. This is one of these timelines reported in the Lancet that shows that about 9 days after the onset of symptoms, is the typical time when an ARDS will set in. And so the time from when an individual is infected to when they show symptoms is somewhere about between 3 to 5 days. So ARDS is being triggered during the second week of infection. And so the big question is, because ARDS is a result of lung damage, then what is actually damaging our lung? Is it the virus or is it our immune system? And so what we've been doing is looking to all of the papers that we can find in the literature that publish time series data in terms of what is happening within the body during the infection. So looking at viral titers, looking at various parameters from the immune system, and trying to get a picture of what is going on that can trigger ARDS or lead to this lung damage. So, and there have been many, many papers on this subject, and I have just pulled individual figures to highlight some of the patterns that have been consistently observed across research groups. And so in terms of viral titers, this time series that I'm showing here is looking at viral titers. The x-axis is going from 0 to 44 days post-symptoms onset, and individuals in green are patients that were categorized as not having severe disease, and then in red are patients that are categorized as having severe disease. And these are viral titers that were, well, these are actually CT values, so this is based on amplification. And these values are representing how much virus is present in the nasopharyngeal swabs. And what I want you to take away from this is that the viral titers are peaking during that first week post-symptoms onset. And by the time you start entering the second week post-symptoms onset, the viral titers are starting to decline. Now, there's not there's missing data early on for the severe patients, but overall, across papers, it has been consistent that the viral titer peaks are happening during that first week post-symptoms onset. And so we know that individuals are ending up on mechanical ventilators and typically ending up in the ICU during that second week. And so this is a time when viral titers are already falling. One thing to take away from this plot is that the severe patients here in red, their titers remain high, so remain above the detection threshold, which is here in blue. They remain high longer than those in green. And that's something that's been seen across multiple studies. So it very well may be the case that individuals who have severe disease have the virus in their body longer, and either that virus could be doing more damage to them and that's helping cause the severe disease, or it could be that their immune system is dysregulated and unable to clear that virus, but the virus itself may not be causing the severe disease. So we've been working on putting information across different parameters along a timeline to figure out what's, just generally get a sense of what's happening within the body. So if we put down this timeline, which is days post-symptoms onset, we have ARDS in that second week, the peak in viral load happening sometime that first week. And now what we really want to think about is the immune system. And so first, if we think about antibodies, so antibodies are typically starting to, I'm sorry, there's a car going by. I live in an area where lots of people play music, so you'll occasionally hear ambulances and music going by. Okay, so if we look at IgG, which is the antibody type that's of interest in terms of neutralizing SARS-CoV-2, IgG doesn't start to be produced until 2 weeks after infection. So this is also during that second week post-symptoms onset. And I can show you one set of plots on this. So these top two graphs are looking at IgG that is specific to the nuclear protein of the virus, and on the left are severe cases, on the right are mild cases, and then the two plots at the bottom are IgGs that are specific to the virus spike protein. But I've put in here these dashed vertical lines, and what that is indicating is day 10 post-symptoms onset, so that's about 2 weeks after infection onset. And you can see that IgG, both the anti-spike and anti-nuclear protein IgG, are starting to kick up around that time. And that's what we expect for infectious diseases in general. 2 weeks is the typical time to launch a novel IgG response. Now, one other thing that's happening during these infections that's very specific to SARS-CoV-2 is lymphopenia and neutrophilia that begins sometime during the first week. And so what is lymphopenia first? So lymphocytes include our T cells, our B cells, and our natural killer cells. And these are cells of our adaptive immune system. Our B cells are the cells that produce antibodies. And the adaptive immune system, it takes about a week or so for these cells to start kicking in and when you have a new infection. Now with lymphopenia, what is happening is that individuals with COVID-19 have been seen to have very low counts of these cells. So their overall lymphocyte numbers are diminished. And just to show you some of these plots and what this looks like, so here on the x-axis I have days from symptoms onset, and then on the y-axis this is the lymphocyte counts. And in red, these are individuals who did not survive the infection, and in blue are individuals that survived. And what I want you to take away from this is in these individuals who died from the infection, even during this, starting from the very first week, they had low lymphocyte counts, and these just dropped further and further and further and further. Now, these are taken from blood samples, and so what's very likely happening is that the lymphocytes that are in our blood are actually honing into the lung. So they're migrating. Cells of our immune system do this all the time. They migrate between our blood and other tissues and organs, and our blood and our lymph nodes. And so what may be happening is we see this big drop in lymphocytes in the blood, but that could be because they're all rushing into the lung to try to deal with the infection and subsequent lung damage. Now, the other thing that is being observed is neutrophilia. So neutrophils are a cell of our innate immune system, and these are cells that kick in immediately when you're infected. And so neutrophilia is being observed during that first week post-symptoms onset in COVID-19 patients. And to show you some data there, again, I have data post-symptoms onset and then neutrophil counts. And in red, these are the severe patients. And so with neutrophilia, it's opposite of lymphopenia. In lymphopenia, patients have too few lymphocytes. In neutrophilia, they have too many lymphocytes. And so we first see a wave, and this is not only reported in this paper, but multiple other papers, where you see high neutrophil counts early on in the infection, and then you have a second wave a couple weeks out. And so we have this, both the innate and adaptive immune system that is responding to this infection and responding kind of in opposite ways. And so another thing that's happening, it's like our bodies just, for individuals who go down the path of severe disease, they're essentially getting multiple hits in terms of what's happening with their immune system. The neutrophilia in the innate immune system, the lymphopenia with the adaptive immune system, and then also they have this cytokine response that starts to kick in during that first week post-infection. And so over this last 4 months or so, there has been a large amount of data published on various cytokines and cytokine responses throughout COVID-19, but there's been one cytokine in particular where there's been very consistent findings across studies and has now started to stick out as being the potentially problematic cytokine. And this is IL-6. And so IL-6 is a cytokine that can act both as a pro-inflammatory cytokine or in other cases, anti-inflammatory. But in COVID-19, it is thought that it is really kicking on inflammation. And so here we have days post-symptoms onset again, and in red, these are the IL-6 levels in individuals who ended up dying of COVID-19. And what you can see is this, not only are their IL-6 levels elevated, but they, throughout the course of the infection, just continue to climb. And the IL-6, the elevated IL-6, especially in severe patients, has been of much interest lately. So the IL-6 is very likely being produced by cells of our innate immune system, so macrophages, dendritic cells, monocytes. But what's happening, or potentially happening, is then that IL-6 is then going and acting upon other cells. And so it is triggering changes in lymphocytes, so it's affecting our adaptive immune system. And then it's also can affect endothelial cells. And so IL-6 is actually very well studied in terms of autoimmunity and inflammatory diseases. So there are several drugs that act to block the activity of IL-6, including one drug for rheumatoid arthritis that is now of interest for COVID-19 treatment. And I'm just going to call this drug TZ because it has a very difficult pronunciation. But with IL-6, what seems to be happening is its production early on in the infection is resulting in this cascade of cytokines and amplification that can lead to systemic levels of inflammation and potentially can be contributing to the lung damage that we see. And the pathological consequences of IL-6, like I said, have been known about for quite some time because it's involved in other inflammatory infections, or inflammatory diseases, I shouldn't say infections, because it's most studied for arthritis. Now, because of this interest in IL-6, there now, actually just as of yesterday, was a paper published in Lancet Rheumatology looking at one of these IL-6 blocking drugs, TZ, to see retrospectively if it increased survival in any patient. So these were data from Italy, and there were several hundred patients who, some of them were given TZ, some of them were given the standard of care. It was not a randomly assigned placebo-controlled trial. It was a retrospective study. But what they show, what these plots are showing are days post-symptoms onset, and then they're looking at the cumulative probability of an individual, this top left one's cumulative probability of an individual dying or being put on a mechanical ventilator if they were given this IL-6 blocker. And the individuals in red are those that were not given the IL-6 blocker. The individuals in blue were given it. And so what you can see, so in the top is whether they ended up on a ventilator or died, and on the bottom is the probability of death. And what they see consistently is that individuals who were given the IL-6 blocker had a reduced probability of the most severe outcomes. And this holds up whether or not they give it intravenously or subcutaneously. So this is hopeful data that by trying to intervene in this cascade that can lead to these cytokine storms, and immunopathology can be one way to potentially treat COVID-19. And so now going back to this timeline, we're starting to get a little bit better picture. So what's happening is first week post-symptoms onset, there's high viral titers. During that same time, you have lymphopenia. So we have all of these lymphocytes leaving our blood, going into the lung, trying to deal with the infection. You have neutrophilia. So we have very high levels of neutrophils. This can be because of proliferation and or a reduced death rate of neutrophils, but the neutrophil population has gotten quite large. And then we also have these inflammatory cytokines, with IL-6 being the primary culprit. And then we have ARDS that kicks in that second week post-infection. And then another thing that has been observed is T-cell exhaustion. So T-cell exhaustion has several definitions. There are actual markers on T cells that can be used to identify whether a T cell is exhausted. And overall, the idea of, or what's believed to be happening in exhausted T cells, is that they have been stimulated so much by the presence of a pathogen that they start to lose their function. And not only can they start to lose their function and their ability to attack that pathogen or respond to the infection, but also can lose their ability to regulate the immune response and harness or the immune response and keep it from getting overreactive. And so this T-cell exhaustion, as I said, it's exactly as it sounds. Our T cells become tired and functionally exhausted. And so in a recent paper in Frontiers in Immunology, it was mentioned that not only do individuals have very low, individuals with lymphopenia have very low T cell numbers in their blood, but for those T cells that are there, they may be exhausted and not capable of doing their jobs adequately. And so this is something really important to think about, especially in the later phases of disease, where the immune system is not only responsible for clearing the infection, but also is responsible for doing its job in terms of homeostasis and healing, that this T cell exhaustion can be problematic. And so because there has been, hold on one second. Waiting for the ambulance to go by. Because there has been so much data that has come out very rapidly over the last several months, one of the things that we did was undergo some data digitization efforts. And so this was led by Sinead Morris, who's a postdoc in Andrew Yates' lab. And so Sinead has been actively, whenever time series are published, well, she either asks authors for data, which we have not had any success in having data shared with us, but in the absence of sharing, going through and hand digitizing all time series that we can get. So to date, she has digitized 1,939 observations, and these cover 27 different parameters that were measured within patients. So things like viral titers, neutrophil counts, T cell counts, IL-6, symptoms, timing. And so just to show you some of the plots we've made from combining data across studies here, and one of the things that's a little bit difficult in terms of trying to bring data together across studies is that oftentimes patient level data are aggregated when they're at the time of publication, and aggregated based on symptoms. And some studies will break down patients in terms of whether they died or they recovered. Others will classify patients as severe or mild, or severe and non-severe, but there overall have not been very standardized clinical definitions in terms of how patients are binned. And so that's been one challenge of trying to bring different data sets together. But across the various studies that we've looked at, here's the data for the neutrophils, and I would just like you to notice the golden time series at the top, which is showing the neutrophilia in patients who died of COVID-19. And this horizontal dash line is showing the normal range, or the upper threshold for the normal range. And so you can see that the neutrophil counts are quite high in individuals who die. And that's consistent with what we had been seeing from individual papers. And the high number of neutrophils that are in the blood is also correlating to high neutrophil levels in the lungs for studies where neutrophil counts have actually been, or neutrophils have been counted in the lung. So overall, the neutrophil population has expanded and they are infiltrating the lung. And one of the reasons why we think that this infiltration of neutrophils in the lung is potentially very important when it comes to lung damage and ARDS is because neutrophils are notorious and known for causing collateral damage to the body. So neutrophils do this really interesting thing where they spew out their chromatin and make these nets that are used as traps to help them deal with pathogens. But these neutrophil nets actually are these extracellular traps, they're called nets. These nets can actually cause damage to the body if they're dysregulated. And so there are several diseases that these nets are known, non-infectious diseases where these nets are known to cause problems, and also infectious diseases. So some of the collateral damage that can be done by neutrophil nets is direct cell damage. They can also delay tissue repair. They can cause inflammation. They can essentially block up your blood vessels. And so the fact that in this first week post symptoms onset and then increasing through that second week, that we have all of this explosion in the neutrophil population, and we know that neutrophils can do direct damage to tissue, suggests that that's one cell type where we really should be looking carefully in trying to understand how is this trauma happening to the lung that then lands people on mechanical ventilators and on this path of potentially succumbing to the disease. Now, I'll quickly go through these other plots. Also in our data digitization efforts, we see the lymphocytes dropping across studies. And one thing that we and also others have found to be particularly informative is calculating the neutrophil to lymphocyte ratio. So in individual patients, if you have neutrophils are shooting up and then you have lymphocytes in the blood are dropping, then calculating that summary statistic of the ratio is quite informative, because the neutrophil to lymphocyte ratio is going to be shooting up extra high if the neutrophils are going up while your lymphocytes are going down. And so these are neutrophil to lymphocyte ratios that we had calculated, with the golden time series being the individuals who died. And so we think that data such as these might be informative for predicting outcomes, and that's something that we would like to explore. And overall, because I want to tie up soon so I can answer questions, but overall, the picture that we've gotten is for these severe patients. As I said, we have viral titers peaking that first week, lymphocytes dropping, neutrophils in yellow going up, and then IL-6 also skyrocketing, that there may be two very critical time points that dictate what path an individual takes. So during this first week post-symptoms onset is when you start to see this initial divergence in terms of people who are going to go down a severe disease path versus non-severe disease path. But then also sometime in the second or third week, there are individuals who are critical, who can be on a mechanical ventilator, be in the ICU, but some of them will go down the path of healing and their lungs being healed or their tissue and healing so that they will eventually get off a mechanical ventilator, versus other individuals that don't trigger that healing process and eventually die. And so we think that, or we propose that there are essentially these two tipping points that should be explored in terms of the within host dynamics and the overall outcomes. And if we and others could potentially find early predictors of what path individuals are going to take, that could be very informative for treatment. And let's see, I wanted to skip through a couple slides. Yep. And so overall, and especially around this time of this second tipping point, where individuals may go down, they've already gotten very ill, they've already have had ARDS triggered and may be on a mechanical ventilator, but though some individuals are going to have the healing process kicked in, which is also a process of the immune system, and while others still have this immunopathology where they're not recovering. And so I don't know if this is the best analogy for this, but the way that I think I'm currently framing this in my mind is it's essentially the good and evil of the immune system, that at some point, once where there's already been all of this immunopathology that's done this damage, at some point, either that healing process is going to win out and the immunopathology is going to get reined in, or it's not. And so I think that this is a really interesting area of research, and if we start looking for some of these early warning signals, it will not only teach us more about the underlying biology, but could also be clinically relevant. And I just wanted to close with saying that understanding the underlying biology in terms of these most severe patients and what's actually precipitating this lung damage and triggering it, and then the processes for healing the lung, is also going to be important for individuals once they've left the hospital. And in recent weeks, there's been a lot of talk in the news about the difficulties of recovering once an individual is off a ventilator, and they very well may be home, but there's been so much damage systemically done to the body, presumably by much of it by the immune system, that we really need to get a handle on really the full breadth of this immunopathology. And I just wanted to highlight a couple recent stories about that. And with that, I will take some questions. I think we have about 10 minutes or so left for questions.
Excellent. Thanks very much, Micaela, for that great presentation. We do have a few questions coming in. I see Andreas has a question. Andreas, I'll go ahead and allow you to talk. If you'd like to go ahead and ask your question and unmute yourself. Yeah.
Well, thanks a lot, Micaela. That was the best overview I've gotten in a while for COVID. So kind of tying in what you said at the end and also at the beginning, and discussions I've had with people, especially when we think about opening colleges in fall, you know, the idea that the younger people might not be that susceptible to death, but the big unknown to me is if they get it and survive, what's the risk that they have long-term damages? And I just haven't seen any good data out there. I've seen anecdotes like you showed just at the end that, you know, people recovering might still have long-term problems, but I'm wondering, have you seen any data?
I haven't, and I think some of that is just also because of the most of the data that's come out in terms of clinical trajectories have been for individuals who are hospitalized and the most severe cases. And so, yeah, I have not seen anything on young people. But one thing in terms of thinking about the immunopathology and age is there's this hyper-inflammatory, I can't remember the exact name of it at this moment, but there's this hyper-inflammatory syndrome that's being observed in children, and it's fairly rare compared to the immunopathology in adults, but that is further suggesting that the immunopathology is really the most important player here. And then also the fact that individuals that have pre-existing conditions for diseases that we know are hyper-inflammatory, like diabetes and obesity, is also contributing. But I haven't seen anything really specific to mild cases in young people.
Okay, we also have a question from Muntaka. I'm going to allow you to.
Mm-hmm. Yep. So the question is that in many papers it was mentioned that IgM is more effective against SARS-CoV-2 than IgG. Can that be cleared? Yeah. So one of the things is IgM. So IgM and IgG are for SARS-CoV-2. I should have shown some IgM plots, but for SARS-CoV-2, they're both produced in that second week post symptoms onset. And the reason that I focused on IgG is because IgG is the isotype that's going to stick around, because IgM is a lot more ephemeral. So it very well may be the case. I haven't looked specifically in terms of IgM versus IgG role, like how effective they each are individually in clearing the virus. But if we're thinking in the longer term, it's going to be the IgG that sticks around and might have the potential for having virus neutralization, months out or years out. So when I think about immunological memory and what antibodies are going to stick around, IgG is the antibody type that we're trying to induce with vaccines and such.
Thanks, Micaela. And I see there's another question in the Q&A box about.
So the second question was, any insights into how long the immunity will last in COVID-19? This is the big multi-billion-dollar question. I won't even call it $1 million question, because right now it's completely unknown. So we do know that immunological memory is kicking in, as I showed with the IgG, production of IgG, and it is antibodies that for all of our, well, I should say all, most of our vaccine preventable diseases, it is antibodies that we're trying to trigger to have that long-lasting memory to be able to go after an infection if you're re-exposed. But the thing is that we really have no idea how long that immunity is going to last. Well, first of all, we don't have an idea if we'll be able to induce sterilizing immunity, which is immunity that will protect you from a subsequent infection. And then we don't know if that kind of immunity is inducible, how long will it last? Will it last 2 months? Will it last 3 years? Will it last your whole life? Is completely unknown at this point. And for some of that, we won't know for many years. We can have vaccine trials that are done within this next year, and especially this fall, there are, I think, something like 18 different vaccines that are competing right now. And we can vaccinate people, let's say now, and have vaccines that trigger a response, are effective for protecting individuals, but those individuals are going to have to live out several years before we can ever find out how long those antibodies are going to last. And so it's a very long game in terms of being able to uncover how long the immunological memory will last. And let's see, so another question. Oh yeah, okay. With these patterns of immune responses, would it make sense to pursue anti-inflammatory plus antibody treatment? So the anti-inflammatory treatment is ongoing, as I had mentioned. Now the antibody treatment if you if you're meaning something like convalescent serum, where you take serum from an individual who survived and has antibodies and giving that to another individual. I know that early on in the pandemic, there were some studies of convalescent serum use, but I don't know how much that has taken off or been used. I don't think that's something that really caught on. If you mean antibody treatment in terms of actually inducing antibodies, like by vaccination, that's something that absolutely, there's going to be work on the vaccines to induce antibodies as preventatives. And then the hope is that also we'll have a therapeutic, which might be these anti-inflammatory drugs. Hopefully some of them will work. Another question, what information do you wish public health departments would disseminate? I was thinking about this yesterday because there has been so much talk about, from the very beginning of the pandemic, about the need to use ventilators and people having respiratory failure. I do think that it would be a great benefit for the public to have some kind of baseline information about how this virus makes us sick and why it is that individuals can be hospitalized for so long. And one of the reasons that individuals can be hospitalized for 40 days, 70 days, is not because the virus is just there, bombarding their body constantly during that time, but that our immune system can be doing a lot of this damage. And I think that the public because the public is very much aware of lots of autoimmune diseases. And so I think that it would also benefit just like being able to understand this pathogen by having some conversation about how our immune system is doing some of the damage.
Also a question from Jeremy. Jeremy, if you'd like to ask your question.
Sure. So I've noted GWAS studies on severe COVID patients versus non-severe COVID patients, identifying a couple of SNPs. Actually looked it up on my genome, I have one of them. And I want to know if there are additional genome regions associated with different immune dysregulation, including the neutrophils and macrophage activity.
Oh, you know what? I don't know, but that's a really interesting
There'll be a subset.
Really interesting question. And I wouldn't be surprised if there are studies out there for GWAS studies for some of these inflammatory autoimmune diseases. So, that could be a really interesting area. If there are certain SNPs that are kind of known to be there in individuals with rheumatoid arthritis or other types of autoimmune diseases, and maybe those might be candidates that you might predict would pop up in some of these immune system dysregulated severe COVID patients. Yeah, that would be a really interesting area of study.
Thank you.
I think we might have time for one more question. So I see Paige also has a question. Paige, you can go ahead and ask your question.
Uh-oh, maybe she's...
Oh, I'm trying to unmute her. Okay.
Hi. I was just wondering about the immunological response within infected COVID patients and any changes in like testing sensitivity depending on their response.
Testing sensitivity in terms of measuring immune system parameters or measuring for virus or antibodies?
Yeah, virus or antibodies.
Yeah, so I haven't seen any studies that have looked at, probability of detecting virus based on what your immune system's doing, but there are certain things that are highly correlated. So for instance, I had shown that plot that in the second during the second week post-symptoms, individuals who are severe cases tend to still have detectable virus around. Those would also be individuals who would have low lymphocyte counts and tend to have high IL-6 and tend to have high neutrophils. So because of those correlations in time, if you were, let's say, just to go grab random people, a subset randomly of individuals in their second week of infection, and wanted to see, can I still pick up virus in this person, then you're going to have a higher detection probability in those individuals who happen to also be having that strong immune response. But it's like a chicken and an egg type of thing, because do they have that higher viral titer because their immune system's doing poorly, or is that high viral titer causing their immune system to do poorly? So all I can say is that because what your immune system's doing and what the virus is doing are highly correlated, that yes, detection probabilities would also correlate with these things. Hopefully that helps or answers it to an extent.
Okay, thanks very much, Micaela. I really appreciate your time and great presentation. That's all we have time for today. Thanks for everyone for attending, and we hope you can join us next month for the next MIDAS webinar with John Drake. So thanks again, Micaela, and thanks everyone, and have a great weekend. See you next month.
Thanks.